I'll just briefly go through the things that I'm going to include in my introduction in the thesis and what data I have at the moment. I hope I think I can get some input before I move to my statistic analysis. This is my final topic. It's about my master thesis, the investigation of small heat shock protein expression under ER stress treated C2C12 mild tubes. And a briefly introduction about the literature. Skeletal muscle basically have a lot of function, and it helps body to perform posture and locomotion and energy metabolism. It store a lot of glucose, and it basically maintain a lot of metabolic pathways. And in aging, basically there has a lot of factors like exercise, nutrition, and hormone regulation that leads to a decrease in general skeletal muscle size. And literature has linked that it will actually has an association with obesity, diabetes, or cancer, this kind of chronic diseases. And to maintain a lot of cell in the body, actually there has a system called proteostasis, which is help protein from unfold protein into a final folded proteins. And in this system, basically you have three main branches, will be protein synthesis and and folding, protein conformation and maintenance, and protein degradation. And this system will basically help most of the cells in the body to throw their 3D structure before they send to their final destination and work and do their function. And in aging, actually, it has been reported that loss of proteostasis is a hallmark of aging, and literature has recently showed that actually loss of proteostasis is linked to some of the muscle-related diseases like sarcopenia and myopathy. And within the proteostasis system, a very important protein called chaperone is basically help to regulate the proteostasis networking system. In general, chaperone has involved in all types of proteostasis network, including assisting protein synthesis. It will also help to block the pathway that will lead to misfold protein aggregates, and then it will help to do protein degradation if the protein is misfold and aggregate. And within the chaperone family, it has a majority of family belongs to small heat shock proteins, which is ATP-dependent heat shock proteins and ATP-independent heat shock proteins. And I'll bring it to later. And of course, some of the non-heat shock protein chaperones family are related. And yeah, now I'm jumped to introducing an organ within the human. It's called endoplasmic reticulum. It's basically at least 2/3 of the proteins are going through into endoplasmic reticulum to regulate, to float the 3D structure properly before they have sent to their final destination. And within ER, it has also the function as protein synthesis, protein folding, and protein quality control. And basically what we know about ER is about proteins. Basically ER's function is basically fold the unfold proteins and send them to final destination. And however, there always have an imbalance of proteostasis or within ER, which is when the amount of proteins need to be folded is more than the folding capacity that ER can be handled. ER stress will be happen, and basically it will occur and a response called unfold protein response and lead to a lot of downstream pathway happening. And the aim of this downstream activation of this pathway is basically try to recover ER into a normal homeostasis condition. Yeah, basically within ER stress or unfold protein response, the first thing that the chaperone does is the bib will dissociate from these three types of UPR-related proteins, and then IRE1 will firstly dimerize and autophosphorylate, and it will happening into the ER, and then it will basically have two types of, in short, basically two types of result. IRE1 dependent decline will be a decrease in mRNA that basically lower the synthesis of mRNA in ER. And then XBP1 is basically a protein that will be sliced in ER and then downstream sent into nucleus and then help to generate some of the proteins that is linked to quality control and which is leading to or some protein related to associated degradation. And for PERK, similar, it will dimerize and phosphorylated and downstream activate another protein called EIF2 alpha. And it will activate another protein, ATF4 is a activate transcription factor. For this protein, it will basically generate CHOP and lead to apoptosis and also leading to some of the amino acid metabolism and autophagy and ER-related proteins that help floating. And ATF6 is the last main branches of UPR pathway, which in the endoplasmic reticulum, it will basically being cleaved into it will send to Golgi apparatus and being cleaved, and then it will cleave once it cleaves, it will downstream into nucleus and also generate some of the increased synthesis more ER-related chaperone or protein, which aim to help to increase the protein floating within the ER. And PDI is basically a chaperone that is around endoplasmic reticulum to do disulfide bond formation of the protein to make them to form into 3D structures. In my master thesis, I will basically dip into very more details about small heat proteins family. And before I jump into the reason or rationale why I studied, I would like to describe in here what small heat protein family does in the proteostasis network. And in general stress condition, basically small heat protein will be activated. And firstly, it's in an oligomerized form. And once it having a stress condition signaling, the small heat protein oligomers will be activated by disassemble from a very big oligomers into smaller pieces of small heat proteins. And then as the stress condition, I'll give an example will be neurodegenerative disease. It will having a lot of accumulation of misfold proteins. And how the small heat protein does is basically they perform a general variholdase function and wait for other small ATP dependent heat protein family to come to deal with the unfold or misfold proteins. This is the general function of small heat protein that we know. And now it will be more specific down to skeletal muscles. As what research or what we know in the literature that has 11 small heat proteins in human genome, and it's only 7 small heat protein exists in skeletal muscles. And I've further go down into the literature and found out that in the Sugiyama's group, they actually perform a yeast-2 hybrid experiment, and they found out that basically HSPB156, they will interact to each other, and then HSPB2 and 3, they will add as an other oligomerase group to perform during misfold protein. And yeah, it would basically jump to basically our DSM's proteomics data, and in his proteomics data, he used an aged comparison between aged skeletal mouse muscle and young skeletal mouse muscle, and he detected in 7 out of 11 small heat proteins in the skeletal muscles, and 4 of them are upregulated. And I found in the literature that basically SAM's proteomics data is some as what literature report in the trend is small heat protein basically increase in the aged skeletal muscles, and it's not only one of the there's a lot of group that mentioned that small Histio protein actually increase in aged skeletal muscles. And same as the ER-related proteins, it's also showing the proteomics data that is increased in the aged mouse muscle. And some of the literature also mentioned that ER-related proteins or chaperone actually increase in aged muscle conditions or related disease. And yeah, this is my hypothesis. And what we know so far is small Histio protein increase in aged muscle, same as the ER-related proteins or chaperones. But actually, no one really studied the direct mechanism between small Histio protein and ER stress. And we just hypothesized that under this condition, will small Histio protein increase under ER stress specific condition as to reduce the amount of mixed protein based on its major function that we know in the literature. And because of that, my master thesis is basically having two chapters. The first chapter will be validation of the antibodies in ER stress conditions and heat shock condition. And the second chapter will be identify the small Histio protein response under different time course and dosage amount of the ER stress drug. And then these are my protein lists and the color, small Histio proteins, pick it, and then UPR pathways, and I highlight in different color represent different pathway that represent and respect colors. And for the last bit will be the classical markers that the literature used it for detecting ER stress, and chaperone or basically just downstream pathway ER markers. And this is the first chapter. I felt the validation of the antibodies in tunica mousin, vepsigargan, and heat shock, and then to show the antibodies are working before I move into the next chapter. And then the second chapter is using different amount of dosage in two types of drugs and induce ER stress in the cells and under different time course duration. And I have high dose and low dose, and then in each drug I use basically 3, 6, 12, 16, 24 hours, and then so one drug will be having high dose and low dose, same as another drug, and all the time courses are the same. And then these are the representative blocks that I have at the moment, and I have still some of the batches need to be imaged and not redo to do it, and I need to wait for a while. And this is the vepsigargan representative block that I have at the moment. As we can see, small Histio proteins 1 and 6, they would basically decrease general under ER stress conditions. Meanwhile, 5 and 7 don't really. And on the right will be UPR stress response figure. And this is the representative figures for the tunica mousin in chapter 2. And it's also show that 1 and 6 small HSPV 1 and 6 decrease over 2 types of dosage treatment in tunica mousin. And UPR response is a bit different in tunica mousin compared to vepsigargan, but this is the figures that I have at the moment. Still, some of them need to be the batches need to be redo, but I need to be wait for a while. And
can you explain what's are the different?
I would say IRE1 doesn't increase in tunica mousin treatment, and ATF6 the click form is different, and EIF2 alpha is different when clinical mouse and compared to Fabsigagan. And yeah, the activation time of different UPR pathways is different between two drugs. And now is the little question that I'm like around my head for these 2 weeks. Basically, I have 12 proteins in total, and now I have 4 conditions, which is high dosage and low dosage in one drug, and I have 2 drugs. So in general, if I really following the figure on the right, one dosage, one graph to represent one protein in one dosage, I'll have basically 48 graphs, 12, 12, 12, 12, which I don't think is possible to do it on my thesis. Yeah, this is the question that I have at the moment, and as long as I got all the figures, and I'll move on to making a statistic graph. And yeah, this is what I'm going to present it today. And yeah, thanks, guys.